The following Torrey talk was given in December 2020 by Natalia Quinteros, a PhD student at the City University of New York and the New York Botanical Garden. Natalia is a grad student of Dr. Doug Daly and is studying spectral plant biology, a topic which she discusses with us in this talk.
I'm going to talk about the field of spectral plant biology and some of my contributions to the field. So this spectral biology is the field in which we investigate the genomic, metabolic, and environmental basis of the variation of plant spectral phenotypes. The plant spectral phenotype or signature can give different kinds of information from the identity of a plant to its physiological status. And this knowledge can help us understand in a large scale how changes in this trait influence biogeochemical cycles and biosphere dynamics. So, but you might be wondering, what is the spectral signature of a plant? So first, let's refresh our memories about the electromagnetic spectrum. So the electromagnetic energy is produced by the vibration of charged particles and have electrical and magnetic properties. The electromagnetic energy travels through a vacuum of space at the speed of light. Our sun is the source of energy across the full spectrum, and it bombards our atmosphere with electromagnetic radiation all the time. The electromagnetic energy travels in waves, and these waves can be very long, like the radio waves. Yeah, like the radio waves that you can see here at the left side of the screen, and or they can be very short, like the gamma waves that you can see at the other end of the spectrum. And our eyes can just detect the visible, the visible part of the spectrum, which is a very short portion of it. So plants receive this electromagnetic energy, absorbs some of it, reflects a portion of it, and also let pass or transmit some of it. So inside the leaf, photosynthetic pigments will absorb the energy in order to carry out photosynthesis, and mostly in the visible range of the spectrum. As you can see in this graph, so chlorophylls absorb most of the energy at around 430 and 660 nanometers, both like in the blue or the red range of the spectrum. And carotenoids and xanthophylls are absorbing energy at around 430 nanometers. But not only photosynthetic pigments are absorbing the incident light. Water, cellulose, and proteins are also absorbing it. So the energy that is not absorbed is either transmitted or reflected back to the environment. And it is obvious to our eyes that there is a strong reflectance in the green region of the spectrum, which gives the leaves the distinct green color. But also at the near infrared region, and followed by the shortwave infrared. And these patterns of reflection give us what is called the spectral signature of leaves. And in the spectral signature, we can recognize areas in which chlorophylls and water are being absorbed, are absorbing this energy inside the leaf. And as you can see from these three species of plants, so the shape and intensity of the spectral signature varies. So, but of course, there is also a response of the spectral signature to the physiological status of the leaf. Here in this diagram, we can see that the reflectance of dead leaf is similar along the whole spectrum. And so that for instance, the stressed leaves we can see both a higher reflectance in the green and the near infrared, which could be indicating that the leaf is lacking some water. And in a healthy leaf, we can see high reflectance in the near infrared part of the spectrum. So this diagram is not showing the shortwave infrared part of the spectrum, because it was to explain, it was designed to explain the patterns that you will see in a sensor that just measures the spectral reflectance from the visible to the near infrared areas of the spectrum. So, and then, so these type of issues needs to be considered when we are planning our work, especially when working in temperate or more seasonal environments, right, where these changes are more noticeable. So when we want to gather information at the leaf level, we use a special instrument called a spectroradiometer, which is equipped with a leaf clip, which allow us to collect spectral reflectance of leaves without the interference of our surroundings. So the spectral reflectance is collected from 400 nanometers, which is in the visible range of the spectrum, passing through the near infrared until almost the end of the shortwave infrared at 240 nanometers. Because it is here where we find features of the leaf that are important for their physiology. And the reflectance intensity is gathered every 1 nanometer. Now, so we can use this information to classify plants at different taxonomic levels. So in this work done in an experimental plot in Minnesota, Anna Schreger and collaborators found that classifying legumes, forbs to their species was more accurate than classifying grasses. But still, in all three cases, with a decent accuracy, if you compare to the fact that in general plants are misclassified 50% of the time in surveys. So the colors in the graphs shows that so for instance in this blue color is showing that, for instance, leaves from the species Sorghastrum nutans were classified as Shuri scoparium, and very often as Anthropogon garardi. So here, right? So Spectral has also helped us to classify species at higher taxonomic level, such as at the family and genus level. This data was gathered at the Enid Hub Conservatory at the New York Botanical Garden, and they are all tropical species from the Rainforest Pavilion. As you can see, species or leaves were classified to their correct botanical families 75% of the time, being more or less accurate depending on the family. In our case, leaves from species belonging to the Myrtaceae and the Rubiaceae were more problematic. At the genus level, the classification was much better, yielding an accuracy of 97%. So just once, a leaf from one of the species of Aphelandra was classified as a Psidium, which is a genus from the Myrtaceae family. So now, with these three examples, we can see that the spectral signature of leaves are good traits to a taxonomist. We can see that classification is better at lower levels, such as genus and species, but there is still not much information about the potential of the spectra to distinguish between closely related species. And that is something that I am interested in. Specifically, I am interested to find what is the power of the spectra to identify closely related species using dry specimens, such as, for example, the specimens that you can find in an herbarium. So, as we saw before, the spectral signature of plants is influenced by their chemical and structural characteristics. So instruments such as optical satellites use these reflectance patterns as informations to derive spectral indices such as greenness, water, and chlorophyll content using the information that we already have about the absorption patterns of these leaf components. So because the satellite is covering a large area, so these spectral indices are giving us information about at the ecosystem level, right? Now with spectral radiometers, such as the one in the bottom, so we can collect reflectance data at much finer scale, and we can calculate the same indices at the leaf level. So we can also use models that are developed to derive more specific parameters based on radiative transfer models. So in a nutshell, a radiative transfer model describes the interaction between the leaf and the incident light. And based on a library of known species and their species traits, these parameters are calculated. So using these radiative models at the leaf level, specifically one called Prospect, we are able to derive traits such as the number of mesophyll layers, right, that give us an indication of how thick that plant is. So chlorophyll A and B density, water content, leaf dry matter per unit of, and finally, leaf dry matter content per unit of area. So compared to the previous indices, which are unitless, here we can get an approximation to chemical and structural components of the leaf. So we use these the spectral reflectance that I tell you guys about before, that we collected at the conservatory to calculate these parameters. And what we found is that the parameter water content is very different in species belonging to the Ericaceae family. So we decided to look closely into these parameters, and here we can see that the water content in Ericaceae is higher than in the other plants. We also compared this derived parameter with the classic water spectral index called normalized difference water index, and we found a similar pattern. And to make sure that these two indices are measuring the same thing, we ran a correlation analysis, and we found that both indices are highly correlated. We did this because radiative transfer models are based on temperate species, and we were using, in this case, tropical species. But as you can see, it works. So then, with the help of a couple of students from LaGuardia Community College, Ludia and Kenya, we analyzed whether the species from the Ericaceae family were statistically different from the rest, and they were. We looked more closely into the origin of the species, and we noticed that the species from the Ericaceae family were mostly from higher altitudes, or from high altitudes, compared to the other species from the other families. Thus, this difference suggests, or this difference in water content suggests something about the natural history of the species. Because as you probably know, species from high altitudes behave similarly to desert species and accumulate water. So now, other parameters such as leaf mass per area can be derived or predicted from spectra using regression models. So in this study by Sean Servin and collaborators, we can see how this trait derived from spectra varies from the Arctic to the tropics. So now, thanks to this contribution, we can use the spectral signature of any plant or group of plants and derive the leaf mass per area without investing time in measuring this trait directly, which takes much more time. Since This study was a methods paper. This type of data allow us to formulate questions regarding latitudinal variation of these kind of traits. So we can also get measures of diversity using spectra as a measure of intraspecific spectral variation. And as we can see in the seminal work of Anna Schreiber and collaborators, this spectral dissimilarity was able to explain the functional distance between species, the phylogenetic distance between species, and even the variation in productivity using regression models as well. Here we can see that the spectral dissimilarity explained 28% of the functional dissimilarity, 20% of the phylogenetic dissimilarity, or distance between species, and 51% of the productivity at the leaf level and 41% at the landscape level. Using optical instruments that capture the spectral reflectance the spectral reflectance of plants from air allow us to cover more area in less time. Instrument mounted in drones and airplanes and satellites allow us for that. Now we can mount several types of instruments that capture the reflectance in specific parts of the spectrum, such as the near infrared and the visible, using 3 bands, the red, the green, and the blue. And also, we can capture hyperspectral images, almost as fine spectral resolution as if we were using an instrument to measure leaf level spectra. And that allow us to have a more finer idea of what's going on with the vegetation at the landscape level. So, for example, with the aid of drones carrying an RGB or a red, green, and blue and hyperspectral sensors, Carol Garzón Lopez and Eloisa Lasso were able to identify 10 paramo species using an RGB sensor with an accuracy higher than 98%, and 4 species of these paramo species with an accuracy of 89%. So paramos are ecosystems which are very high up in the Colombian mountains and which are not of easy access. We can use instrument mounting in drones to map the leaf economic spectrum, which is basically the trade-off between the leaf mass per area, nitrogen, phosphorus, and leaf photosynthesis. For instance, in this work from Eleanor Thompson and collaborators, show us different areas of West Africa in which we can find different types of tropical forests based on the quantities of these essential nutrients. They also found that these trade-offs were correlated with precipitation and substrate, where these plants are growing. So this was all done with a regression model. And the resolution that we can get is so good that this information is very valuable for local land management and conservation efforts. So now we can also obtain very important information of forestry diversity for larger areas using small airplanes, carrying, for instance, these laser-guided spectrometers. So Greg Asner and collaborators covered an area of 72 million hectares in the Peruvian Amazon and generated a suite of forest canopy functional traits. This data was used by the Peruvian government to optimize decision-making regarding areas of higher threats. So, and also here in this paper, they were able to find out, so what in the landscape was generating these patterns of variation in each specific functional trait, right? If it was either the geology, the elevation, the slope, the hydrology, etc. So, but not only functional traits can be derived from airborne spectral data. So since plants differentially interact with solar radiation, so plant spectral diversity is an integrator of plant chemical structural and taxonomic diversity that can be remotely sensed. So Etienne Liberté and his collaborators have proposed that we can measure this diversity as spectral variance, which can be partitioned from regional or gamma diversity into within community and among communities component or additive alpha and beta components. So with all this information, and thanks to the Torrey Botanical Society Training Fellowship, I am participating in a project with data collected at the Rocky Mountain Biological Laboratory in Colorado that looks into integrating classic measures of taxonomic and functional diversity with spectral reflectance at the leaf, plot, and landscape levels to better understand the changes in spectral diversity in an altitudinal gradient. So finally, with this classic type of information, so this graph shows us the classic information that we gather or we use to advance the theoretical and applied biological, ecological, and conservation sciences, right? We use the accepted taxonomic names, we use trait data, we use genetic data, and geographical data. So now, if we add the spectral data, so we could be contributing to a deeper understanding of some of these factors, right? So because the spectral data again contains information regarding the taxonomy and the functional component of the leaves. And with this, well, I went very fast through my presentation, so we're gonna have a lot of time to discuss things. I want to thank the Graduate Center at CUNY, the New York Botanical Garden, and the Torrey Botanical Society, my advisor, Dr. Daly, all my thesis committee, and other students who have collaborated with their input and ideas to my my own project. And also to you, thank you very much for listening. And now, so I open the floor for questions.
Thank you very much, Natalia. That was a really interesting presentation. Everybody, I'm just going to reiterate that if you have questions, feel free to write them in the chat. I will read them to Natalia, and she'll answer them for you. So while you guys figure out if you have any questions for the speaker, I have one or two. Actually, I have a lot. Because this is a really interesting topic, and I'd love to learn more about it. So I'm curious about the methods that you use for the handheld scanners, right? You go leaf to leaf and scan them like that. And I'm very interested in the amount of that. This is super interesting because I imagine that from a leaf to leaf basis, there's probably a lot of variation, and I saw that you sort of alluded to that as well. You discussed that a little bit. I was wondering how many replicates you usually have to take, scans of a particular species to get its sort of profile, I guess.
Yeah, no, that's an excellent question, Jordan. So usually what we do is so it is depending of the question, right? So for species classification, so like most of the publications have used from 3 all the way to probably like 5 leaves per individual. So you actually can get, as you can see here, so well, in this data set, there were many individuals per species, right? Each color line represents a species. So you can see that inside each leaf, so, or between the leaves, so you can find a lot of variability. So and we use again from so some like preliminary, some old work have used from like 2 leaves, and so for our study at the conservatory we actually use 5 leaves per species.
And do you just scan a small point on the leaf, or is it more of a like, I don't know how the machine works exactly.
Yeah, no, that's again an excellent question. So the way that we actually measure is, so well, it depends of the size of the leaf, right? So they're species that they have a very big leaf blade, right? So what you want to get is a sample of around or the area of the leaf,
Mhm.
right? So, but what you want to avoid is the main veins, right? Because
Uh,
in order to get, like, for example, parameters such as the number of mesophyll layers, right? So you don't want, you don't, or the amount of chlorophyll,
mhm.
right? You don't want to hit these very big vascular bundles.
Okay. Amy Burkhoff says, I think it looked like in Peru there was an inverse distribution of N and P. An explanation?
Let me. Yes, so as we see here in the previous work that that I show from this African environment, so we can see that you can find trade-offs between these components, right? Like the leaf mass per area, the amount of nitrogen, right, which is N in the leaf, and the amount of phosphorus. So so that so and you can find it so in different amounts. So that depending probably of the substrate where the forest is growing, right? It could be a soil issue, right? Because as you guys probably know, so nitrogen, phosphorus, and potassium are like the essential elements that plant use for their metabolism.
Mhm.
I don't know if that answered Amy's question.
Uh,
Yeah, it's interesting. I didn't see that when I read the paper.
Sandra has a question. I'm sorry if I missed it, but it seems like Natalia said that that she wants to use spectroscopy on herbarium specimens, but it seems like water content is an important distinguishing trait. Can Natalia discuss any more about results on herbarium specimens and what traits are important?
Yes, so there are actually ways to infer the amount of water from the from a dry specimen. So this is a work that we are just starting. So we want to use so the idea of so using so if you let's see what is this. Let me find the cartoon very quickly. So if you see in a dead leaf, so we can still see some a higher reflectance in the near infrared with respect of the visible, right? So we think that we can still get some variability, like enough variability between um, the spectra to be able to classify the species. And so, and that's part of the question, right? So so we are also so part I am also doing a test on so how much resolution we lose when from like a dry like a fresh specimen to a dry specimen, right? And that's something else that is um, a project that is happening and it's being a little halted, you know, due to the whole pandemic situation. But yeah, no, it's you know, those are things that um, we want to see. Like first, like how much information we're losing um, from like fresh to dry spectra, and also if with dry spectra. Um, so we can still tell apart the species.
Okay. Dean has a question. Do these methods work well with grasslands or shrublands?
So in which term, like identifying species or getting functional traits, or both?
We'll wait for Dean to respond to that, but both.
Okay, so um, I've seen publications about shrublands, and they seem to work well, both to infer like functional traits and identify species. So I don't remember exactly the publication, but for grasslands, I haven't really seen anything regarding grasslands, to be honest.
Okay. Howard has a question. Are your reflectances all done on the upper leaf surfaces? Any done on the lower leaf sides? And if so, any patterns upper or lower surfaces?
Yeah, that's an excellent question. So we are just measuring uh, the leaf reflectance at the top of the leaf, and so usually what happens is like if you see the instrument, right? So so the light is coming from the top. And so we usually do this because most of like if you think about this type of dicot plants, so the palisade mesophyll, it's going to be the one that is containing most of the chlorophyll, right? And also, if we see here in this cross section of the leaf, like what is happening on the other, like what we will be capturing on the other side of the leaf is mostly like a lot of like air spaces, right? And so I think you can expect, if I remember correctly, so like most of the work that is done is usually just by measuring the reflectance on the top of the leaf or the adaxial part, right? So and not that much in the bottom, but yeah, you can probably expect a different pattern. And also in most, if you see most of the leaves, the coloration of the top part is always much darker than in the bottom of the leaf.
Okay. So to kind of piggyback on who was it? Was it Dean's question? I was curious if there has been any research like this on desert systems, because I feel like aerial scans of desert habitats using like satellite or aerial spectroscopy would be a pretty good way to scan large swaths of otherwise miserable stretches of desert for plant life and identify it and stuff like that. I know that you discussed the similarity between montane habitats and desert, or montane plants and desert plants. But I was curious if you knew of anything in desert habitat.
Yeah. So, well, if you guys are interested, there is this network of ecological observatories called NEON, right? Like as the light, right? So so they actually are measuring, so they have especially here in the United States, so they have many ecological observatories in different type of ecosystems, and they have gathered data from like leaf level all the way to airborne. So if anybody in the audience is interested in these type of ecosystems, I'm sure that NEON has that type of data, and usually they're freely available for anybody who wants to do research with them. But to be honest, I haven't seen any. I'm mostly interested in tropical moist forest. So
Right.
so yeah, no, I haven't read anything regarding.
Yeah, it falls
but it's a very good
without
question.
it falls outside of your focus. Yeah.
Yeah, exactly.
We got a few more questions here. Caesar asks, can you still differentiate between genera across species in different families that have convergent morphologies?
Yeah, so this is so what many papers suggest is that, so yeah, you can, the thing is like spectra, well, if I understand your question correctly, Cesar, so spectra will look beyond the, let's say the leaf shape, right? So supposedly there is like intrinsic characteristics of the species that are gonna be registered in the spectral signature that allow us to classify species into their genera and species level. Because if you think about it, so like for tropical species, right, which are mostly broad leaves, right, or non-deciduous broad leaves. So it's like the spectra still works, right? To identify the species or to classify the species into the genera. At least that's what we've been, and also to the species level in that, like other people have found.
Okay. We have another question from Howard. We find that in most plants, reflectances are greater from the lower surface, but if a leaf is vertically oriented, like cattails, we find no differences. We are trying to figure out the ecophysiological difference of the pattern and whether there is a phylogenetic signal. Your talk has been interesting and relates to our work. Enjoyed your talk. Okay, not so much a question, just a really interesting comment. Thank you very much, Howard.
So could you repeat the first part of the comment?
Yeah, so Howard says that they find that in most plants, reflectances are greater from the lower surface, but if a leaf is vertically oriented, like cattails, for example, they find no differences.
Mm-hmm. Oh, that's very interesting. And they were looking for phylogenetic signals, right?
Yes.
So actually, I didn't mention this, but we have run some phylogenetic signal analyses on the spectral signature of the species that we collected at the conservatory, and we found a very low signal in like certain areas. I think it was, wait, I don't recall right now. But something in which we actually like found some signal was when we derived leaf mass per area. So we found some like low, but still some phylogenetic signal on that trait. So that was interesting. But there is other work on phylogenetic signal, and it's been, it's been hard to really to find a strong signal, especially at the species level. It seems like at higher orders, it's easier to find that phylogenetic signal.
Really interesting stuff. So we're gonna kind of wind down here. If anybody else has any last questions, get them in. I have one more question for you.
Mm-hmm.
So I was curious about whether spectroscopy. You kind of, one of your figures showed that there is a difference between stressed and non-stressed leaves,
Mm-hmm.
and I was curious if there is a difference that is detectable to like sort of measure and observe the growth and eventual senescence, or sometimes like maybe even like suffering pathogens, like these sorts of life cycle situations that plants go through, if these are detectable through spectroscopy.
Yes. So actually, yes.
Differentiate.
So if you think about the life cycle of the leaf, right, depending of the species, so like the amount of let's say pigments, right, inside the leaf is different than to a mature leaf, right? So also the structural characteristics. So like newer leaves are sometimes much softer than an older leaf, right? And everything. So all these characteristics are picked by the spectral signature. So so in the type of work that I've been doing, so we've been trying to use the same type of leaf, so a mature leaf that is sunlit, right? That has like all the chlorophyll that needs, right?
Right.
Like it has reached maturity, so it has all the structural components. So that way we can, like we can compare, right, between between species, right?
Hmm.
So yeah, no, definitely you're going to find a difference in different stages of the leaf, and you can pick that up even, you know, at the when you're gathering spectral data from a satellite.
Interesting. Do you think that like the difference between a a senescing leaf and a diseased leaf is something that is uh, distinguishable by spectroscopy, or is that not really known by you yet, or like has been studied?
So yeah, that's something that well, I haven't really measured,
Hmm.
but I will expect that, yeah, you will see. So actually, there was a very interesting study about the implications of like moving the samples,
Hmm.
right, from the sampling side to, let's say, to like a lab, right, and how much. So how much of how much this movement was going to cost in terms of the quality of the spectra. And so what they found is that with time you're going to start seeing a drastic change on the spectral signature of the leaf, and that's because of the natural, you know, decay of the tissues. And also when, you know, like, let's say you're entering into fall, right, in temperate areas. So like just the movement of the pigments, right, are gonna create also a change on the phylogenetic signal.
Yeah, that makes
I'm sorry,
sense.
not phylogenetic signal, the spectral signature.
Yeah. Yeah, that makes a lot of sense. Okay, thank you.
Yeah.
So this looks to be the end of questions. So I'm just gonna, on behalf of everyone here, I'm gonna say thank you very much for this great talk, Natalia. Thanks
Oh, no, thank you.
for coming to speak to us like this. This is a very cool topic.
Yeah.
Um, so I'm just
Hopefully in the
gonna,
near future we'll have more more information to share when
yeah, that would be
I'm able to
fantastic.
respond.
Yeah, when your work is all ready and everything, and you have lots of stuff to report, we're gonna have to get you back on here and
Yeah, that'll be awesome
give us the update.
for sure.